Hi students, it is happy to me once again to meet you to discuss about the lesson. Yesterday we were talking about, that is what we have, the structural aspects related to the testis, ovary, etc. along with the mammary glands. Now we have to pass on to the next one, the process of formation of gametes. That is called gametogenesis. That's the next part. What is gametogenesis? I mentioned already genesis. Genesis means formation, development, birth, etc. So the process of formation of male and female gametes from the primary sex organs, namely the testis and ovary, and that is called gametogenesis. So the gametogenesis is very important because the continuity of life depends only on the sex cells, namely the sperm and the egg. During gametogenesis, we have meiotic division, that is reduction division. So the meiotic division plays a significant role during gametogenesis. Normally, this is a common word, gametogenesis, formation of simply gametes, either sperm or egg. Now let's start with the formation of the male gametes. The formation of the male gametes, namely the sperm, that is called spermatogenesis, formation of male gametes or sperm. So the sequence of events that occur in the seminiferous tubules of the testis to produce the sperm is called spermatogenesis. The sequence of events that occur in the seminiferous tubules of testis that produce the sperm, that process is called spermatogenesis. Now during development what will happen? We have the primordial germ cells. Primordia, the base, primordial germ cells. That is nothing but the ancestral cells. The primordial germ cells migrate into, that is what we have the testis, and become modified into immature germ cells called the spermatogonia. So we have primordial germ cells migrate into the testis and become modified into what we have the germ cells called spermatogonia or sperm mother cells. That's the first stage, primordial germ cells. The next one, what we have the germinal cells called spermatogonia, other is called sperm mother cells. Now the spermatogonia, the process of actually spermatogenesis or the mitotic division of the spermatogonia begin at the age of what is called puberty. So first we have the formation of initial cells, what are called the spermatogonia. So they undergo mitotic divisions at what is called puberty and continue the same process, that is a mitotic division, throughout the life. So male is more fertile, having a longer duration of fertile than the female. So the fertility of the female starts before on or before 50th year of age, whereas in the case of spermatogenesis or the formation of sex cells or the fertility of the male, just it goes on, just even up to the age of 70 or 75. Okay, now just we have the sperm mother cells. What are called the spermatogonia? Just coming from the primordial germ cells, continue to divide just at the age of puberty or at puberty and continue the same process throughout the life. So that's why indicated is seen that one spermatogonia divide by mitotic divisions, producing more and more spermatogonia. It's a continuous process, mitotic division, or what is called spermatogonia. That's why we put two arrow marks, division, to produce more and more spermatogonia. Now what is the first stage? So in the first stage of development, the spermatogonia, what we have the sperm mother cells, migrate along with what we have the Sertoli cells to the central lumen of the seminiferous tubules. Central lumen of the seminiferous tubules, where some sort of growth occurs and become enlarged and modified into what is called a primary spermatocyte with a diploid number of chromosomes. So both spermatogonia and primary spermatocyte which are diploid in nature. Okay, the primary spermatocyte after a relative period of growth gets converted to primary spermatocyte. I am taking only one just spermatogonium. It becomes modified, enlarged form, primary spermatocyte. It is diploid number. Now the process of mitotic division of the spermatogonia to form more and more spermatogonia together constitute what is called the multiplication phase. Multiplication phase. There is nothing but the division of the sperm mother cells of spermatogonia to form more and more number of spermatogonia. Now I mentioned about just each spermatogonium becomes modified and enlarged. This is because of a little growth. That is called as a growth phase to become the primary spermatocyte, which is 2n number. Now the primary spermatocyte enters into a maturation phase. So now we have the next one, maturation phase. So number one, multiplication phase, increase number of spermatogonia. Growth phase, a slight increase in size of the spermatogonial cell to form primary spermatocyte. Now the next one, the primary spermatocyte undergoes a period of what is called maturation. The maturation is nothing but what we have meiotic division 1 and 2. So that what will happen, the chromosome number of primary spermatocyte gets reduced to haploid. Okay, so each primary spermatocyte which is diploid undergoes the first meiotic division to form two equal cells with n number of chromosomes, and that is called secondary spermatocyte. Two cells, secondary spermatocyte, each one with haploid number of chromosomes. You know, in a meiotic division, the first division is a reduction division, the second one is a normal modified mitotic division. Now each primary spermatocyte enters into the second meiotic division, so that each one gets converted into two spermatids. Altogether, there are 4 spermatids are produced from a single primary spermatocyte. You can see that one. From a single primary spermatocyte, we have received actually 4 non-functional sperms called the spermatids. The spermatids are spherical structures, spherical cells with n number of chromosomes. They are also considered as a non-functional sperms. Non-functional sperms. So from each primary spermatocyte we have received, that is 4 spermatids. Suppose 100 spermatids are produced. How many primary spermatocytes are needed to form 100? The formula is n by 4. Suppose the number of spermatids 100, because from each what is called primary spermatocyte 4 spermatids are produced. So 100 by 4 we have what is called 25 divisions are needed to form what is called 4. That is what we have actually, I am telling that one, 100 spermatids. To form 100 spermatids we need 25 divisions. Okay, or we can say not 25 divisions, we can need what is called 25 primary spermatocytes, because from each one, that is, we have 4 spermatid cells. So the spermatids are non-functional sperms. Now the non-functional sperms have a nucleus with a haploid number of chromosomes. They enter into the next period, what is called differentiation period. The process of differentiation is called spermatogenesis. The whole process called actually, that is spermatogenesis. Now the next one, spermiogenesis. The process of conversion of spermatid into functional sperms, that is spermatozoa, and that is called spermiogenesis. So spermatogenesis is a whole process. Spermiogenesis, also called by name spermiotelosis, that results in the formation of what we have, the sperms. So each spermatid undergoes differentiation to form what is called actually a spermatozoa. The sperms is an abbreviated form of spermatozoa. This is the plural form, spermatozoan singular. So we have 4 spermatozoa with the haploid number of chromosomes produced from a single what is called primary spermatocyte. Okay, that's why I mentioned the formula. N by 4, 100 by 4, we have 25 primary spermatocytes are needed to form 100 spermatids. So the 100 spermatids undergo differentiation to form what is called the spermatozoa with n number of chromosomes. Okay, so the process of conversion of spermatid into functional sperm is called spermiogenesis. We'll continue further. Here I represented the sequence of events that happen during spermatogenesis. We have the primordial germ cells 2n. By mitosis, they gave rise to what is called the spermatogonia. By repeated mitotic division, once again we have received primary spermatocytes along with their little growth. Now the primary spermatocyte enters into, that is the first meiotic division, that is nothing but the maturation process to form two haploid secondary spermatocytes. Now primordial germ cells, spermatogonia and primary spermatocyte all are diploid in nature, whereas the secondary spermatocyte is haploid in nature. Now, which undergoes a second meiotic division to form, that is what is called spermatids. That is, from each what we have, the primary spermatocyte, 4 spermatids are produced, all are haploid. By differentiation, namely spermiogenesis process, now what is happening, the spermatids get converted into the functional spermatozoa, which are also haploid. Now, the sperms once formed, they are released into the central lumen of the seminiferous tubules. The process of release of sperms into the central lumen of the seminiferous tubules is called spermiation process. What is spermiation? The release of sperms into the lumen of the seminiferous tubules. The release. Now the whole process of spermatogenesis occurs about in 64 days. About 64 days. If you are analyzing the testis at a given time, the different regions of seminiferous tubules contain spermatocytes in different stages of development. At a given time, if you are analyzing the nature of the testis or seminiferous tubules, the different regions of the seminiferous tubules contain what we have, the spermatocytes in different stages of development. Now normally the sperm production is nearly at a constant rate. For example, per day, 200 million sperms are produced per day. 200 million sperms. So per day. So nearly at a constant rate, the production of the sperm, that is what we have, 200 million sperms are produced in male per day. So the whole process of spermatogenesis normally takes place in 64 days. The release of the sperm into the central cavity or lumen of seminiferous tubules is called spermiation. Now the next one we have to go for the hormonal control of spermatogenesis. The process of spermatogenesis is regulated and controlled by hormones of different regions. Now, spermatogenesis begins at the age of puberty due to, actually it is initiated, we can say initiated due to the increased release of what we have, that is gonadotropin releasing hormone. So the process is initiated due to increase in the release of gonadotropin releasing hormone from hypothalamus. So GnRH releasing hormone. Now these hormones or factors act on the anterior pituitary to release two hormones to control the activity of testes. Number one, follicle stimulating hormone, otherwise called FSH. Follicle stimulating hormone, FSH. What is the role of FSH in, that is spermatogenesis? It is responsible for the growth of the testis, or we can say testicular growth. At the same time, it also enhances the production of one protein, androgen binding protein, that is simply called ABP, by the Sertoli cells. Now androgen binding protein, this is normally produced by the Sertoli cells, the nurse cells, you know that one. So which in turn stimulate the process of spermatogenesis. So testicular growth, otherwise another one, enhances the production of androgen binding protein by the Sertoli cells, which in turn stimulate the spermatogenesis. This is one hormone. Now another hormone, what we have, the luteinizing hormone, it's also called by another name, interstitial cell stimulating hormone, LH or ICSH. as it stimulates the interstitial cells, otherwise called the Leydig cells. Interstitial cells are otherwise called Leydig cells, hence the name interstitial cell stimulating hormone. This hormone normally acts on the Leydig cells or the interstitial cells to release the male sex hormone androgen. One such male sex hormone is testosterone. The testosterone is responsible for stimulating the process of spermatogenesis along with FSH. So both FSH as well as testosterone together augment, that is the process of spermatogenesis. That's about the hormonal control. One gonadotropin releasing hormone and then two hormones by the anti-pituitary both work together and stimulate the process of what we have, the spermatogenesis. Okay, so we also have some inhibin. I mentioned the hormone inhibin. Inhibin is also secreted by what is called the Sertoli cells. This hormone inhibits the activity of FSH. We discussed already. So, inhibin is secreted by what we have, Sertoli cells. It also secretes androgen binding protein. Androgen binding protein is a stimulator of spermatogenesis, and inhibin is an inhibitor of spermatogenesis, suppresses the process of spermatogenesis. So with that I concluded the spermatogenesis, the various events along with the pictures. We have to discuss the structure of sperm and oogenesis in the next class.